Welcome to the deep dive. Today, we're jumping into something, well, pretty fundamental to, like, all life.
Absolutely.
How cells, especially plant cells, take simple glucose and turn it into the energy that runs, well, everything.
It's this amazing, complex, but also really elegant process.
Right. So our mission today is to kind of unpack that. We're drawing from the Raven biology of plants, specifically the chapter on glucose oxidation.
And what's fascinating, I think, is just how universal this is. We're connecting this simple sugar molecule to the energy that drives a flagellum's flick or pumps stuff across membrane.
And it all comes back to ATT, right? This universal energy currency.
Exactly. ADP, it powers biosynthesis, movement, transport, you name it.
So we want to give you a shortcut to understanding how things like sucrose or starch get converted into ATP. Let's peel back the layers. Okay, so let's start big picture. Cellular respiration. What is it, fundamentally?
At its core, it's the complete oxidation of sugars, or other organic molecules, breaking them down into carbon dioxide and water to release energy.
But it doesn't just start with glucose floating around, does it? Especially in plants.
No, usually you have storage molecules first, like starch or sucrose. Those need to be hydrolyzed, broken down into simpler sugars, like glucose or fructose. Glucose is usually our starting point for respiration itself.
And oxidation, that means losing electrons, right?
Precisely. Glucose gets oxidized, it loses electrons and often hydrogen atoms along with them. And something else has to gain them.
Which is usually oxygen in the main pathway.
In aerobic respiration, yes. Oxygen gets reduced, it gains those electrons, and combines with protons to form water. It's this transfer of electrons from a high-energy state in glucose to a lower one in water that releases the usable energy.
But it's controlled, yeah, not like setting glucose on fire.
Exactly. It's a series of controlled steps. And the conditions matter. You can have aerobic respiration with oxygen or anaerobic without it.
And aerobic gives the biggest energy payout.
By far. The overall equation tells the story. 1 glucose plus 6 oxygen gives you 6 carbon dioxide, 6 water, and a release of about 686 kilocalories per mole. That's the maximum potential.
Wow. Okay, and this doesn't happen all at once. You mentioned stages.
Right. 4 main stages for the aerobic pathway. First, glycolysis. Second, forming acetyl-CoA. Third, the citric acid cycle. And fourth, the electron transport chain, coupled with oxidative phosphorylation.
And where does most of this action happen?
In eukaryotes, it's mainly inside the mitochondria, if you picture maybe like a jelly bean. It has an outer membrane and then a very folded inner membrane. Those folds are called cristae.
Cristae, okay.
And that folding is key because it creates a huge surface area packed with the enzymes needed for the later stages, especially ATP production. It's the cell's power plant, basically.
Got it. So the powerhouse. Let's dive into stage 1 then, glycolysis. Sounds like sugar splitting.
That's exactly what it means, glycolysis. You take one 6-carbon glucose molecule and split it into 2 3-carbon molecules called pyruvate.
And where does this happen? Inside the mitochondria?
Nope, this first part is actually out in the cytosol, the main cell fluid. And critically, it's anaerobic.
No oxygen needed.
No oxygen required for glycolysis itself. And that's important because almost every living cell does glycolysis. It suggests it's, a really ancient pathway, probably evolved before Earth even had much oxygen in its atmosphere.
Wow. Okay, so 10 steps, you said. How does that work? Is it just chop?
Not quite. It's more strategic. Think of it in 2 main phases. First, there's the preparatory phase, or energy investment phase.
Investment. The cell has to spend energy first.
It does. It uses 2 ATP molecules early on. These ATPs add phosphate groups to the glucose, kind of activating it, making it unstable and ready to be split.
Okay, so add phosphates, rearrange it a bit.
All right, it becomes fructose 1,6-bisphosphate. Then that 6-carbon molecule splits into 2 different 3-carbon molecules, one of which quickly converts to be identical to the other. So you end up with 2 molecules of glyceraldehyde 3-phosphate.
2G3P, okay. And we've spent 2 ATP.
Exactly. Now comes the payoff phase. This is where the cell gets its energy back and more.
All right, payoff time.
So those 2 G3P molecules are oxidized. Electrons are removed and captured by an electron carrier molecule called NAD plus ERA, reducing it to NADH. You get 2 NADH molecules total here.
NADH. That sounds important. Like carrying energy.
It is. It's carrying high-energy electrons that will be cashed in later for more ATP if oxygen is around.
Okay, keep going. Payoff phase.
And then, through a couple more steps, phosphate groups are transferred directly from the intermediates onto ADP molecules to make ADP. This happens twice for each G3P molecule, so 4 times total.
4 ATP made directly.
Yep, that's called substrate-level phosphorylation because the phosphate comes directly from a substrate molecule. So you made 4 ATP, but you spent 2 earlier.
So a net gain of 2 ATP.
Exactly. The net result of glycolysis is 2 pyruvate, 2 NADH, and 2 ATP per glucose.
Okay, 2 ATP net. That doesn't sound like a huge amount compared to the 686 kilocalories we talked about.
You're absolutely right. That's a key point. Most of the original energy from glucose, maybe 80% or so, is still locked up in those 2 pyruvate molecules and the 2 NADH. Glycolysis just cracks the door open.
Right, it's like step 1. So this raises the big question.
What happens next? How do you get the rest of that energy?
Yeah.
And the answer depends entirely on whether oxygen is available.
The fork in the road. Let's take the oxygen path first, the aerobic one.
Okay, so if oxygen is present, those 2 pyruvate molecules get transported from the cytosol into the mitochondria, specifically into the inner compartment called the matrix.
Into the matrix. Okay.
Once inside, pyruvate undergoes a really crucial reaction before the main cycle starts. It gets oxidized.
Oxidized again.
Yes. 1 carbon atom is removed and released as carbon dioxide CO2. That's the first time we see CO2 being produced.
Ah, okay. So a 3-carbon pyruvate becomes something with 2 carbons.
Exactly. Electrons are also removed, reducing another NAD+ to NADH. And that remaining 2-carbon fragment, called an acetyl group, gets attached to a carrier molecule called coenzyme A.
Forming acetyl-CoA.
Acetyl-CoA, exactly. And since we started with 2 pyruvates from 1 glucose, this whole process happens twice. So you get 2 molecules of acetyl-CoA, 2 NADH, and you release 2 molecules of CO2.
Okay, quick recap. Glucose 6C to 2-pyruvate 3C in glycolysis, then 2-pyruvate 3C to 2-acetyl-CoA 2C in the mitochondrial matrix, releasing 2 CO2 and making 2 NADH.
Perfect. Now those 2 acetyl-CoA molecules are ready for the main event in the matrix.
The citric acid cycle, also called the Krebs cycle, right?
Yep, or the TCA cycle. All the same thing. It's a cycle, meaning it starts and ends with the same molecule.
How does it start?
The 2-carbon acetyl group from acetyl-CoA joins up with a 4-carbon molecule already present in the matrix, called oxaloacetate.
2 carbons plus 4 carbons gives 6 carbon.
That forms a 6-carbon molecule called citrate. That's why it's the citric acid cycle. Coenzyme A is released at this point, free to go pick up another acetyl group.
Okay, we have citrate. Now what? It's a cycle, so we need to get back to that 4-carbon oxaloacetate.
Exactly. Through a series of enzyme-catalyzed steps, that citrate molecule is progressively oxidized. 2 more carbon atoms are removed and released as CO2.
2 CO2 per cycle turn.
Per turn, yes. And remember, we have 2 acetyl-CoA entering per glucose, so that's another 4 CO2 released here in total.
Wait, so we had 2 CO2 from making acetyl-CoA, and now 4 CO2 from 2 turns of the citric acid cycle. That's 6 CO2. Is that all the carbons from the original glucose?
That's it. By the end of the citric acid cycle, all 6 carbons from the starting glucose molecule have been completely oxidized and released as CO2.
Wow, okay. But where's the energy? We got 2 ATP from glycolysis. Did we get more here?
We get a little bit directly. Each turn of the cycle produces 1 molecule of ATP by substrate-level phosphorylation. So that's 2 more ATP per glucose.
2 more ATP. Okay, so 4 total so far. Still seems low.
But the main energy capture in the citric acid cycle isn't direct ATP. It's capturing high-energy electrons. Each turn generates 3 molecules of NADH and 1 molecule of another electron carrier called FADH₂.
FADH₂, similar to NADH.
Very similar. It also carries high-energy electrons, just at a slightly lower energy level than NADH. So for 2 turns of the cycle per glucose, you get a total of 6 NADH and 2 FADH₂, plus the 2 direct ATP.
Okay, so after the citric acid cycle, the glucose is gone as CO2. We have a net of 4 ATP directly, but now we have a whole bunch of these electron carriers. 2 NADH from glycolysis, 2 NADH from making acetyl-CoA, and 6 NADH plus 2 FADH₂ from the citric acid cycle. That's 10 NADH and 2 FADH₂.
Precisely. And that's where most of the energy originally in glucose is now stored in those energized electrons carried by NADH and FADH₂.
So the final stage must be about cashing in those carriers, right?
Exactly. That's the job of the electron transport chain, or ETC.
Look at it where? Back on those inner membrane folds, the cristae.
Yes, embedded right within the inner mitochondrial membrane. The ETC is a series of protein complexes and other electron-carrying molecules.
Like a bucket brigade for electrons.
Kind of. The NADH and FADH₂ deliver their high-energy electrons to the beginning of the chain. Then, the electrons are passed sequentially from one carrier to the next, moving downhill in terms of energy level.
Like a waterfall. Each drop releases energy.
A great analogy. As electrons move from a higher energy carrier to a lower one, they release free energy, and the cell cleverly harnesses that energy
To do what?
to pump protons, hydrogen ions, H plus bond. Specific protein complexes in the ETC use the energy released from electron flow to actively pump protons from the mitochondrial matrix out into the intermembrane space.
The space between the inner and outer membranes.
Correct. So you're building up a high concentration of protons in that space relative to the matrix.
Pumping protons out. And what happens to the electrons at the very end of the chain? Where do they go?
This is where oxygen finally comes in. Oxygen is the final electron acceptor. It's sitting at the bottom of the energy waterfall, waiting. It accepts the now low-energy electrons and combines with protons from the surrounding solution to form water, H2O.
Ah, so that's why we need to breathe oxygen, to clear out the electrons at the end of the chain. Without it, the chain would back up.
Exactly. Everything would grind to a halt. No electron flow, no proton pumping.
Okay, so we've used electron energy to pump protons out, creating this high concentration in the intermembrane space. How does that make ATP?
This is the genius part, called chemiosmosis or chemiosmotic coupling. That buildup of protons creates an electrochemical gradient. It's like water behind a dam's stored potential energy.
A proton dam. Okay.
And embedded in that same intermembrane is a remarkable molecular machine called ATP synthase.
Sounds like it makes ATP.
It does. It has a channel that allows protons to flow back down their concentration gradient from the intermembrane space back into the matrix. It's the only way back for them.
Like water slowing through a turbine in the dam?
Precisely. And just like a turbine uses water flow to generate electricity, ATP synthase uses the flow of protons moving through it to power the synthesis of ATP. It takes ADP and inorganic phosphate, pi, and smashes them together to make ATP.
Wow. So the electron transport chain sets up the proton gradient, and ATP synthase uses the energy of that gradient flowing back down to make ATP. That's oxidative phosphorylation.
That's oxidative phosphorylation powered by chemiosmosis. Oxidative because it depends on the oxidation of NADH and FADH₂ and ultimately oxygen, and phosphorylation. Because we're adding phosphate to ADP. It's incredibly efficient.
How efficient? What's the final tally? We had 4 direct ATP. How many from all those NADH and FADH₂ going through the ETC and chemiosmosis?
Well, the exact number can vary slightly depending on conditions and how things are shuttled, but the generally accepted yield is about 3 ATP per NADH that goes through the full chain and about 2 ATP per FADH₂.
Okay, so 10 NADH times 3 is 30 ATP, and 2 FADH₂ times 2 is 4 ATP. That's 34 ATP from oxidative phosphorylation.
Roughly, yes. But remember, the NADH from glycolysis happened outside the mitochondria.
Yeah.
Getting their electrons inside sometimes costs a bit of energy, depending on the shuttle system used. So the typical net maximum yield from one glucose molecule under aerobic conditions is usually calculated as 36 ATP.
36. Okay, that's way more than the 2 from glycolysis alone.
Immensely more. About 18 times more ATP.
Yeah.
It represents capturing around 38% of the total energy available in glucose, which is actually remarkably efficient for a biological process. The rest is lost as heat, which isn't entirely wasted. It helps maintain body temperature in many organisms.
So 36 ATP, that's the power of oxygen.
That's the power of aerobic respiration.
Now, you mentioned earlier that cells aren't just stuck with glucose. Can they burn other things for energy using these same pathways?
Absolutely. Fats and proteins can also feed into this system. It's very flexible.
How do fats work?
Fats, or triglycerides, are broken down into glycerol and fatty acids. Fatty acids then undergo a process called beta oxidation, where they're chopped up into 2 carbon units.
2 carbon units. Sounds familiar?
Yep, they become acetyl-CoA, which can then enter the citric acid cycle directly. Fatty acids are very energy-rich. A single long fatty acid can produce a lot of acetyl-CoA and thus a lot of ATP.
And proteins.
Proteins are broken down into amino acids. The amino group, the nitrogen part, is removed, and the remaining carbon skeletons can be converted into various intermediates that enter the process at different points. Some become pyruvate, some acetyl-CoA, some directly enter the citric acid cycle as intermediates like alpha-ketoglutarate or oxaloacetate.
So the system is like a central processing hub that can accept fuel from different sources.
Exactly. It's highly integrated.
Okay, but what happens if the oxygen isn't there? We took the aerobic path, but what about the anaerobic fork in the road?
Right. If oxygen is scarce or absent, the electron transport chain backs up because oxygen isn't there to accept the electrons at the end.
No electron flow, no proton pumping, no oxidative phosphorylation.
Correct. And crucially, the NADH and FADH₂ produced in glycolysis and the citric acid cycle can't get reoxidized back to NAD+ and FAD by the ETC.
Why is that a problem?
because glycolysis needs NAD plus to accept electrons in the payoff phase. If all the cell's NAD plus gets stuck as NADH, glycolysis stops.
Ah, so even that initial 2 ATP net gain shuts down.
It would, unless the cell has another way to regenerate NAD plus from NADH. And that's what fermentation pathways do.
Fermentation. Okay, like making beer or yogurt.
Those are classic examples. There are two main types we usually talk about. First, lactate fermentation.
Lactate, like lactic acid in mussels.
Exactly. In this pathway, pyruvate itself accepts the electrons from NADH, becoming reduced to lactate. This reaction oxidizes NADH back to NAD+, allowing glycolysis to continue producing its small amount of ATP. Many bacteria, fungi, and our muscle cells during intense exercise do this.
So you get 2 ATP from glycolysis, and the pyruvate just turns into lactate to keep glycolysis going.
That's the gist of it. The second type is alcohol fermentation.
The beer and wine one.
Right. Common in yeast and most plant cells under anaerobic conditions. Here, pyruvate first has a carbon removed as CO2. The resulting 2-carbon molecule, acetaldehyde, then accepts electrons from NADH, becoming reduced to ethanol, ethyl alcohol.
So you get ethanol and CO2 as products, and again, the NADH gets turned back into NAD plus D.
Precisely. So the net equation for alcohol fermentation is glucose yields 2 ethanol, 2 CO2, and 2 ATP from glycolysis. For lactate, it's glucose yields 2 lactate and 2 ATP.
In both cases, only 2 ATP net gain. That's it.
That's it. Much, much less energy captured compared to aerobic respiration, only about 7% of glucose's total potential energy. Most of the energy remains locked in the lactate or ethanol molecules.
But it allows life to persist when oxygen isn't available.
Critically important for survival in anaerobic environments. And, as you mentioned, hugely important culturally for humans using yeast for baking and brewing for millennia. That Botany of Beer example really highlights it, converting starch to sugar, then yeast ferments the sugar to alcohol and CO2.
It's amazing how these ancient pathways are still so relevant. Glycolysis being anaerobic makes sense if it evolved early on.
It certainly suggests that. It's the most fundamental, widespread energy-releasing pathway.
So pulling it all together, these pathways aren't just about breaking stuff down, are they? You mentioned intermediates being used elsewhere.
Yes, that's a crucial concept. We talk about catabolism pathways that break down molecules to release energy, like we've been discussing. But cells also need anabolism pathways that build complex molecules.
Building proteins, fats, etc.
Right. And the intermediates from catabolism, like pyruvate, acetyl-CoA, and molecules within the citric acid cycle are often the starting points, the precursors for anabolic pathways.
So the citric acid cycle isn't just burning fuel, it's also like a supply depot for building materials.
Exactly. It's a central metabolic hub connecting breakdown and buildup. That's true for heterotrophic cells, which get their organic molecules from outside, and even for autotrophic cells, like plants doing photosynthesis, which integrate these pathways with their own sugar production.
That really changes how you think about it. It's not just a one-way street to ATP.
Not at all. It's a dynamic network.
Okay, so let's recap the big takeaways from this deep dive. We started with glucose, this basic sugar.
And saw how glycolysis, out in the cytosol, splits it into 2 pyruvates, giving a small net gain of 2 ATP and 2 NADH without needing oxygen.
Then, if oxygen is present, pyruvate enters the mitochondria, gets converted to acetyl-CoA, releasing CO2 and making more NADH.
That acetyl-CoA then enters the citric acid cycle in the matrix, where the remaining carbons are fully oxidized to CO2, generating a little more direct ATP, but importantly, lots of NADH and FADH₂.
And those electron carriers then shuttle their high-energy electrons to the electron transport chain on the inner mitochondrial membrane,
where the flow of electrons powers proton pumping, creating a gradient across the membrane.
Which then drives ATP synthase, using the energy of protons flowing back down to produce the vast majority of ATP, about 36 total per glucose aerobically. Chemiosmosis.
Compared to just the 2 ATP gained via anaerobic fermentation pathways, like lactate or alcohol fermentation, which are essential for regenerating NAD+ when oxygen is absent.
And we saw the mitochondria as the powerhouse, the citric acid cycle as a central metabolic hub, and the flexibility of the system to use fats and proteins too.
Right. Understanding these core processes really shows the underlying unity of life, how cells manage their energy with incredible efficiency and adaptability. It's quite remarkable.
It really is. So maybe a final thought for everyone listening. Consider the sheer elegance of it all. These intricate, sequential biochemical steps conserved across vast swaths of life. What does that intricate optimization tell you about the power of evolution in shaping how life captures and uses energy from something as simple as a sugar molecule? We hope this deep dive into glucose oxidation gave you some real aha moments and a clearer picture of how life powers itself. From the entire team, thank you for joining us on this deep dive. This has been a warm thank you from the Last Minute Lecture team.